So, good morning or good afternoon, everybody. It's my pleasure to continue our global IMMUNOTALK series with another highly distinguished speaker, Shabaana Khader. And I'd like to briefly introduce her to you. She's an internationally recognized expert on tuberculosis. Her research delves into host pathogen interactions and TB infections with a particular focus on granulomas, where we know that granulomas are those structures where bacteria are contained, where they can survive for long periods before they eventually leave and cause a massive pathology. She has made really key and highly significant contributions in understanding the mechanisms that regulate those processes and that regulate the escape or that cause the escape of bacteria from these granulomas. Beyond this, her work extends into vaccine development, diagnostics, treatments of TB, and here she collaborates with academic and industrial partners. She's also very interested in teaching and promoting awareness for topics such as vaccine development, vaccine hesitancy, pandemic preparedness, how climate change impacts microbial diversity, and of course into the key topic of antibiotic resistance, which is also highly important, of course, for TB infections. Now, Shabaana holds a PhD in biotechnology that she obtained from Madurai Kamayai University in India. I hope I pronounced this somewhat correctly. She then held faculty positions at the School of Medicine of the University of Pittsburgh and at the University of Washington in St. Louis, where she was a professor in molecular microbiology. And as of 2022, she has become a professor or chair of the Department of Microbiology at the University of Chicago. Now she's also a fellow of the American Academy of Microbiology, and of course I could continue for a long period highlighting your research, but I will leave this up to you, and I would say once again, thank you very much for accepting to be our speaker for today, and we look forward to your research. And I'd like to start our meeting with usually the opening question that is dedicated to young people, to newcomers into faculty positions. And here my question is, if you look at your career, what is an advice, what were key decisions that you have made in your career that influenced your career, and what advice from there would you give to young people who start an academic career? Thank you very much for being here.
Thank you, Dietmar, Elena, for inviting me. This is obviously such an impactful platform for interaction across the world, and it's really an honor and privilege to be here today. Dietmar, in response to your question, this is something I tell my trainees all the time, because I think we come from so many different backgrounds, so many different cultures. We have very diverse interests, diverse expertise. And one of the things that I've tried to do, and I tell this to all of my trainees who are in the lab and who've been in the lab, is to really bring your authentic self, be true to who you are while we navigate many different spaces and we learn to fit in. I think by staying true to ourselves, whether it's our science, our expertise, it really allows us to grow and always continue to be who we are. And that I have always found is my core anchor. Things change around you, expectations change, but if you are true to who you are, you kind of keep that anchor that allows you to move forward. So that's been one of my guiding values as I've navigated my own career. I think you're muted.
So turn on my microphone. I think that's an excellent advice, and I think it's really important. Yes, fully agree. Well, now it's up for you to share your slides, and as I said, we look forward to your presentation.
Thank you. Can everybody see my slides and they look okay? And everybody can hear me fine, I hope. I don't know if there's a live chat, but I hope everyone can hear me as well, that I'm loud enough. So really what I thought I would do today is share some of our data, some of the experiments we've done to really understand the lung landscape in the context of tuberculosis, and give one or two small examples of how we've used that information to move our discoveries really from the bench to the bedside, with the hope of translating the findings that we've done in the lab to making new vaccines and discoveries for tuberculosis vaccines. As Dietmar mentioned, I just recently moved my lab from Washington University in St. Louis, where I was for 10 years, to the University of Chicago to take on the role of the chair of the Department of Microbiology at University of Chicago. I do have some disclosures. I have collaborative research agreements or contracts with some of the organizations that are described here: Blue Willow Biologics, IDRI, Adjuvants Technologies, J&J Biologics. And one of the vaccine platforms I will be talking about is a patent that I co-own with Blue Willow Biologics. So those are my disclosures up front. I think for this audience, a global infectious disease such as tuberculosis probably does not need very elaborate introduction, but I know we have very diverse interests here. So to make the point really that tuberculosis continues to be one of the top infectious disease killers in the world. Before COVID and post-COVID, it is the largest infectious disease killer. For example, in 2023, we had 1.3 million TB deaths, and this is the leading cause of death among people with HIV co-infection. Obviously, of those 1.3 million people who are dying from TB, a good 500,000 of those cases are from multi-drug resistant TB cases. But herein lies the problem, right? We have 2 billion people, about 1/4 of the world's population, that is thought to be latently infected with Mycobacterium tuberculosis. So you have a large reservoir of infected people who go on to progress to active clinical disease. So as you can see here, the statistics are about 1.6 million people go on to get clinical disease every year from that huge reservoir of people that we have. So clearly TB is a disease that is difficult to control. It's been with us for thousands of years and is one that continues to kill at the highest level. So just to bring the focus back to what my lab does, I'd like to anchor you to the central core circle here, which is where really describes the work that we do. The work that my lab does uses lots of in vitro model systems, including both ex vivo as well as organoid model system. We use in vivo model systems such as the small animal models. As you know, there's advantages to using those. They're very tractable animal models, but there are also limitations to using the small animal models. So really we use both of these in vitro and in vivo models to then probe and take some of our science and discoveries from these models to a slightly more translational preclinical model of macaque model of TB, where you really see the spectrum of tuberculosis that you see in human tuberculosis. And then finally, what we have done in many of our studies is link off all of our discoveries in the bench to the human tuberculosis to really understand if our discoveries translate to biomarkers or understanding host immunity to tuberculosis in human tuberculosis using samples. And as I will show in a couple of slides, our collaborations are across the world, really in a global sense. And then if you look at the second ring of what we really are trying to achieve is use all of these model systems and our discoveries to help us inform two main things. One is we really want to understand the host immune response to tuberculosis that's highlighted here on the top. And the second, a very important part of that same intersect of the other side of the coin is how is MTB interacting with the host immune cells and how is it advocating for its own pathogenesis? And if you now look at the outer circle there, you'll see two highlighted, you'll see two things that we're really focusing on. By doing all of this really allows us to understand the disease pathogenesis of tuberculosis. And we use all of this information to our ultimate goal, which is really making TB vaccines. One other point, as we go through my slides today, you will see these little icons come up in your on my right-hand corner of the slide. So that'll tell you which species is the data that I'm showing you from, just to orient you, because I will be showing you data across many different species. And what will help you is the little symbol at the corner to tell you what species specifically I'm talking to you about. So moving on from here, I actually like to do my acknowledgements up front. And as I just told you, I just moved my lab from WashU to University of Chicago, and some of the people who moved with me, which I'm very fortunate for and thankful for. But certainly we have now had to rebuild our team at University of Chicago. And this was a picture taken about a couple of weeks ago, where we had a celebration, potluck celebration, to celebrate a lot of different things, including student and papers, grants, etc. So I'm really grateful for all the people who moved and the new people who really trusted the group to rebuild our science again. And you will see I will individually thank people as we go through our projects. And as I mentioned a little bit before, I'll also like to say this up front and acknowledge that the work that I'm going to show you today is a true collaboration in many ways. It is, in my own mind, a way that team science really succeeds. As you know, we are at the University of Chicago, but really all of our collaborators and the data that I'm going to share with you today is work we've done with folks across the world at multiple institutions, within the US, but also national. If you look internationally, in the University of Mexico, there's Dr. Joaquim Zuniga, or we work with Subhash Babu in India, as well as several of our collaborators are in South Africa. And obviously, I've been very, very grateful that with this team partnership, we've been able to get and maintain some level of funding that's enabled us to keep this team science moving at the level that we are very happy with. So the two objectives that I'm going to really talk about today is really, as Dietmar mentioned, I'm going to talk about what do we understand about the human TB granuloma. What are new insights that my lab has provided and helped move the field forward? How can we use that now and talk about some two different novel strategies that we're using in the lab to kind of move this towards a translational angle in the context of TB vaccine development? So those will be the two objectives that I will be focusing on in my presentation today. So coming back to the core disease pathogenesis, as I said, TB is an infectious disease. 2 billion people are infected. TB infects the lower airways. 90 to 95% of people who get exposed to the infection are in fact having a very, very good immune response to the infection. They can control the infection asymptomatically, even though they carry the infection. They do not have any clinical symptoms of the disease. Only about 5 to 10% of the infected people then go on to get clinical active disease. But as I mentioned, the problem lies herein. You have 2 billion people who carry the infection, and over their lifetime, they have a 10% risk of going on to get clinical disease. And the reactivation that we talk about here could be due to a variety of different reasons. It could be COVID co-infection, it could be HIV co-infection. We know that worms drive TB to progress towards. So it could be a variety of different factors that allow this latently infected population to now progress towards active clinical disease. And of course, the TB patients who have active clinical disease are the people who are now transmitting the cycle of transmission to continue the cycle of infection. So if you look classically at the lung TB granuloma, this is actually a TB granuloma from a mini pig from one of Larry Schleisinger's reviews. But this is very similar to the structure you would see in a human TB granuloma. So this is classic textbook knowledge of what we say a granuloma looks like. It has a central necrotic core of macrophages. TB could be intracellular. Within the necrotic core, certainly TB could be extracellular, and people imagine that we have this cuff of lymphocytes that surround, and usually the thought is that it walls off TB from now infecting other sites. But certainly we know this information because we've looked at active TB granulomas from patients, and we know that this is the late stage disease granuloma. But what we really don't understand, and one of the early things that my lab really wanted to understand, is what happens in those 90 to 95% of infected people who are actually controlling infection? Meaning, what is the difference between a granuloma that's protective in the context of latent TB, and what happens in the context of moving towards disease progression, that is the states that we call active TB? Obviously, I'm simplifying it. There's a whole spectrum of subclinical TB between latent TB and active TB. That, for the purpose of my talk, I will not be talking about. So we started out about, I want to say 10 years ago, where a graduate student in my lab, Samantha Slide, really now looked at human biopsies. And again, please follow the code to tell you what species I'm talking about. So these samples are from human active TB lung biopsies. And she started really looking at these lesions to see if there were other cell types apart from the classic granuloma, where you see this necrotic core and this lymphocyte cuff. What she was really looking for was to see if there were structures that we now call tertiary lymphoid organs, or we can also call them as inducible bronchus-associated lymphoid structures, which in humans are usually very close to a bronchus. So what Samantha was able to find is in some of these human TB lesions, she found that the cells do form certain structures that look like lymphoid structures. For example, here you can see CD3 staining in red. You can see the presence of follicular dendritic cells shown here in green, and the presence of IgD right within those follicles. You can also see B cell proliferating cells. You can see PCNA staining in red, and you can see that the B cells are proliferating by staining for CD20. You can also see the presence of T follicle helper cells shown here by CXCR5 staining on CD3 cells, or ICOS staining shown here in green within those lesions. Another feature that is important when you call a structure a tertiary lymphoid structure is really the presence of homeostatic chemokines such as CXCL13, CCL19, and CCL21. And Samantha was able to show that whether you look here by mRNA in situ hybridization, or whether you look by protein expression, that the expression of CXCL13, which is a key homeostatic chemokine, is expressed within these lesions. But here was the surprise. We kept scoring multitudes of human TB lesions, and what we found was these structures were only found in a very small proportion. I want to say about 30% of the human active TB lesions actually had these structures. So Samantha came up with the hypothesis, perhaps what we are looking at is a protective feature of TB, and therefore it's not reflected during clinical disease. And so the hypothesis was that we would see well-represented follicles in the lung if we were to use a model where we could look at both TB latency and we were able to look at active clinical disease. And for that purpose, we really collaborated at that point, one of our very early collaborations with Deepak Kaushal's lab, where he has a well-documented model of macaque model of TB, where he can infect the monkeys and get either latent TB, so these monkeys seroconvert and are PPD positive post-infection, or these monkeys go on to get active tuberculosis and have all the clinical symptoms of TB. So going into this model, Samantha's hypothesis really was that in active TB, you'd see the classic necrotic TB granulomas, but the structures that she called then as eyeball structures or lymphoid follicles would be well represented in the context of latent TB. And so we got samples from Deepak Kaushal's lab, and here's the lung, which you can see here, H&E sections right at the left side corner of the slides. You can see when you look under the lung, Samantha's hypothesis was absolutely correct in that the lungs do have these large necrotic cores if you have clinical TB, just like you would see in human tuberculosis. Macaques, however, that had latent TB, so we knew they're infected. We knew we couldn't get any sputum, any culture positivity for TB, but however, we knew these monkeys had converted, seroconverted by PPD testing. What she found in the lungs of these monkeys were, in fact, these little tight follicles. When you stain for CD20 and PCNA, which is proliferating marker, you can see these highly dense areas of lymphoid follicles. And if you do the same staining in the lungs of macaques with active tuberculosis, you do not see those structures. You can certainly quantitate both the inflammatory rate, inflammation in the monkeys, as well as quantitate the B cell follicles. And that data is shown here. So essentially, just as you would speculate, the inflammatory areas shown here by the large necrotic areas is highly well reflected in monkeys with active tuberculosis. But the opposite, that is the formation of these tight lymphoid follicles, is actually a feature of protection that we show in the context of latent tuberculosis. So since then, we've done a lot of work in this space, really understanding what are those immune cells that are there, really deep diving into a variety of different chemokines and chemokine markers, as well as shown that IL-17 and Th17 cells are important for forming these structures that we now call as granuloma-associated lymphoid tissue or eyeball structures, depending on the species we are looking at. And obviously, the work that we've done here has allowed us to say that these structures are protective in the context of tuberculosis, and forming these structures is one way to continue to form protection during latent tuberculosis. But certainly at that point, being at WashU, it was a perfect opportunity to really ask the question, what was the role of innate lymphoid cells in driving these early structures? And this was work we did in collaboration with Marco Colonno's lab. In fact, we just published a paper earlier this week, also with Marco's lab. And we asked the question, is there a role for the innate lymphoid cells, such as ILC1s, ILC2s, and ILC3s, in seeding or driving these responses in the lung in the context of infection? As many of you may know already, the innate lymphoid cells are really the counterparts on the innate lymphoid side. They are driven by similar transcription factors as the Th1, Th2, and Th17 counterparts, For example, TBET drives ILC1, GATA3 drives ILC2, and ROR gamma T drives the ILC3 subsets. So really the hypothesis that we went in asking were these ILC3s in any way driving the seeding or the initial formation of these lymphoid structures. And this was work that we did both across the human studies as well as in mouse models of tuberculosis. This particular slide, as you can see, is from human tuberculosis. This was work that was done in Al Leslie's lab in South Africa. And what his student, Amanda Arden, at that time did was really measured the frequencies of ILC1s, ILC2s, and ILC3s in the peripheral blood. And you can see that TB negative are healthy controls. But in the context of tuberculosis, whether you look at HIV negative or HIV co-infected individuals, you see that during tuberculosis, ILC3s, ILC1s, ILC2s, and ILC3s are in fact depleted in the periphery. And in this paper that we published in 2019, we actually showed that these cells migrate to the lung in the context of human TB infection. So obviously now we wanted to look at the mechanisms of protection, whether ILCs were mediating protection. And two outstanding postdocs in my lab spearheaded this work, Shibali Das and Raquel Domingo Gonzalez. And what they did was infected the mouse model of tuberculosis with low doses of MTB infection and then tracked the number of ILC1s, the number of ILC2s, and the number of ILC3s that come into the lung post-infection. And what they were able to show was really remarkable, very, very early infiltration or accumulation of ILC1s and ILC3s, particularly post-infection. There were baseline higher levels of ILC3s that continued, ILC2s that continued to increase, but really the accumulation of ILC3s at day 5 is much earlier than any other innate cell type that happens in the lung post-infection. So both Shibali and Raquel then went on to show that when you infect a RAC2 common gamma chain knockout mice, so essentially these mice lack all T cell, B cells, but also lack all of the ILC components, including NK cells. They were able to show that when compared to a RAG1 knockout mice, which lacks both T cells and B cells, these mice are extremely susceptible to MTB infection. And you can see that when we measure the log 10 CFU MTB per lung, these mice are more susceptible. Certainly, they were able to rescue the susceptibility simply by adoptively transferring back ILC2s. And just to show you the data again, you can see that the RAC2 common gamma chain knockout mice did have a deficiency in ILC1, ILC2, and ILC3 post-infection. Since this is published data, I'm not going to show you all the data. You have to take my word for it. What they were able to show that it was likely not an ILC1 defect that was causing this increased susceptibility or ILC2 defect. Using a variety of tools that Marco Colonno's lab had generated, we were able to show that specifically it was the ILC3 population and specifically their role in driving IL-23 and IL-17 that was important in this very, very early impact in control, because the data that I'm showing here really is day 15 post-infection. This is a time point before which T cells and B cells actually accumulate in the lung in the context of MTB infection in the mouse model. So going from here, very recent data, in fact, as recent of just earlier this week, Shibali Das went on to show in her next paper that if you sorted out ILCs from the lungs at day 5 or day 14 post-infection, you'd see a variety of subpopulations of ILCs in the lung if you measure them by single-cell RNA sequencing. Certainly, you can see the key feature is that ILC3s accumulate by day 14. You can see there is an ILC3, ILC1-like subtype as well. And if you look at the markers that these different subpopulations of ILCs in the lung are expressing, certainly the ILC3, ILC1 subtype is expressing CXCR3. They're certainly expressing transcription factors such as RUNX3. They're certainly expressing XBT16. RORC is very uniquely expressed in ILC3s. TCSF7, which is a transcription factor for lineage of ILC3s, is expressed across ILC3s and ILC1s. Again, ILC3s express CCR6. So we're really learning a lot about the early accumulation of ILCs as a subset early in the context of forming these structures following MTB infection. So really, at this point, just to summarize what I've said, we now know that ILC3s are one of the early cells that respond to MTB infection. We've also shown that they make IL-22 and IL-17. that induces chemokines such as CXCL13. And we've published other studies where we've knocked out CXCL13 or CXCR5 and shown that, in fact, this makes the mouse very susceptible to MTB infection. And this is because they do not form these structures. We've also shown other studies that the IL-17, IL-22 coming later from Th17 cells is an important feature that continues to keep these structures to be formed and do the function that they need to do, which is really bring the T cells and the B cells to interact with the infected macrophages to continue to kill MTB infection within the context of these follicles. So since this is all published data, I'm just going to leave this slide at here and then move on to the next part of it. So what I showed you just now was the story where we went in with a hypothesis that perhaps ILC3s were important for control of TB and then tested the hypothesis because we had the right tools to do it. In this next few slides that I'm going to show, we did the opposite, which is we said we want to go in an unbiased way and use unbiased analysis and do deep sequencing of the lung. And here we looked at control macaques, we looked at monkeys that had active TB, and we had monkeys that had latent TB. And this was work that was spearheaded by 3 people, Kapia, who was in Max Artyomov's lab at WashU, Shibali Das in my lab, and Dheeraj Singh, who was in Deepak Kaushal's lab. And so we compared across the 3 different stages, which is control, latency, and active tuberculosis. And we looked at just unbiased, what is different in terms of the lung landscape. If you look at it by either sRNA-seq or CyTOF, and then we did validation studies both in humans as well as non-human primates. And I'm just, since again, this was published a couple of years ago, I'm going to hit on just the key points and the high notes of the paper. One of the really curious things, at least for me, that really came up was very, very dramatic increase in NK cells, which is another type of innate lymphoid cell. And this was really increase in the context of latent tuberculosis. And we could do this by sRNA-seq, we could do this by CyTOF. And really, if you look at the transcriptional profile, you're looking at the blue here at the corner, which is latent TB. You can see that these are classically expressing effector molecules of NK cells, such as perforin, granzyme, as well as granulysin. So this is a classic increase in expression of accumulation of cells, but also expression of effector molecules in the lungs of monkeys that are able to control infection. So obviously, as I promised you, we use the human model to kind of validate our studies that we see in the lung of monkeys to the periphery of human tuberculosis. So these are patients who have latent tuberculosis or active tuberculosis, and we measured the frequency of NK cells in the PBMCs, and then also measured the increase in the number of cells when you restimulated these cells with MTB. And you can see that latently infected individuals in the periphery have higher levels of NK cells, and they do respond much better upon stimulation with TB. Certainly, when we sorted these NK cells from patients from either healthy controls or latent TB, we were able to reconfirm that even in human tuberculosis, that these NK cells in the periphery express higher levels of granulysin and granzyme, but not so much interferon gamma and IL-22. So just to put this back in the model I've been talking to you about, we've shown now that ILCs, including ILC3s and NK cells as an innate lymphoid cell, are necessary or perhaps play an important early role in seeding these structures that are important for control of MTB infection. And certainly, one of the things we've noted all along is the presence of B cell within those structures. And I've already shown you that the T cells within those lymphoid structures express classic markers of T follicular helper cells, such as expression of CXCR5 and ICOS. So the next question we really focused on was what was the role for B cells and what was the role for T follicular helper cells in the context of forming these structures? And to do this, 2 outstanding postdocs in the lab, along with a technician of mine who really generated a lot of the tools to look and ask this specific question. And the question they asked was a relatively simple question, which is what do those B cells do in the context of MTB protection? And is there a role specifically for TF8 cells? But this would require us to make a variety of different tools, which Lan Lu, one of my technicians, was able to generate these different mouse lines. So essentially, she generated a mouse line that lacked CD19, where she crossed the CD19 CRE with the IRF4, and she also crossed the CD19 CRE with the BLIMP1. So these mice would not form antibodies in the context after the crosses had been made. She also generated mice that lacked B cells. And so these are CD19 Cree mice that lacked IL-10 production. So these are B regulatory cells. They also generated mice that lacked germinal center B cells by crossing the CD19 Cree with the BCL6 flux. And then finally, we had mice that did not express class 2 expression on B cells. And of course, mice that had non-specific B cells. In this case, these mice would only recognize a hen lysozyme and not recognize TB. And so really using these different conditional knockout mice, both Rosemary and Ananya went down a journey, but they really wanted to know if any of these B cell effector functions were important for either formation of those lymphoid structures or protection in the context of MTB infection. And finally, they generated a mouse that lacked TF8 cells by generating a cross between CD4 Cree and BCL6 floxed mice. The first piece of data is shown here. And this data is really showing that if you take a mouse that has non-specific B cells, so it has the entire repertoire of B cells, but the B cells do not recognize MTB antigen. They recognize a hen lysozyme antigen. You will see that these mice, as you infect and look at a low-dose infection model in tuberculosis, these mice become more susceptible as infection progresses, shown here by day 75 post-infection and day 100 post-infection. But here comes the surprise. And certainly when we knocked out any of these genes that are effector functions in B cells specifically, we did not see that they induced increased susceptibility in infection in any of these knockouts. So essentially, we are looking at mice that lack plasma cells or germinal center B cells, or knocking out class 2 expression on B cells, antibody production, and certainly knocking out IL-10 from B cells did not seem to have an impact on increased susceptibility to infection. So this suggested to us that B cell specific, MTB specific B cells are necessary, but independent of classical B cell effector functions in the context of MTB infection. So they had to dig in a little bit deeper. One of the first things they did is ask if T follicular helper cells were important, or even if germinal center B cells were important. And so they generated the appropriate crosses. And as you can see here, T follicular helper cells are important very early in the context of infection. Certainly, these mice die about 100 days post-infection. You can see they have about 7 logs of bacteria. And if you measure and see this, do not form these structures. We are calling it granuloma-associated lymphoid structure here. And you can see that mice that do not have T follicular helper cells, shown here in the next slide, in the next panel here, they do not form these lymphoid structures. So clearly, at least you're convinced by this data that T follicular helper cells are important. B cells are important. However, the B cells are not really important in the context of the classical effector mechanisms of protection that we think about B cells to do. So after a lot of work that both Rosemary and Ananya did in this study, what they were able to show was, in fact, the B cells are playing a very important role in localizing the T follicular helper cells within those structures to really mediate MTB killing. And that data is captured here in this one slide. So if you look at the gray area that's under an arbitrary threshold, you'll see 3 mice strains that are below this threshold. The first one is the CD4 Cree IRF4 floxed mice, the CD4 Cree BCL6 floxed mice, and the MD4 mice, which are the MTB non-specific mice. Those are the only strains of mice that are unable to localize T follicular helper cells. By knocking out any other B cell function, you still get localization of B cells. And so this tells us that really the crucial function that B cells are doing here is interacting with the T follicular helper cells. And Rosemary and Ananya were able to show that this was through a PD-1, PD-L1 interaction. And they were localizing and drawing in the T follicular helper cells to form these structures where the MTB protection was going on. And so really at this point, we know that B cells mediate the TF8 cell localization within the lymphoid follicle. And here's an example of really looking at that very visually, where you see these outlined areas are the follicles. What you're seeing here is the 16S RNA staining for MTB in brown. So you can see that wherever these follicles are actually areas of exclusion where TB is really not propagating very well until very late into infection. So forming these structures may be one way that we can actually accelerate and improve vaccine design for tuberculosis. And so far what I've told you is what's happening in the lung in the context of latent TB. And with the interest that if we can understand that, maybe we can design vaccines that we can now use that knowledge to design new vaccines for tuberculosis. But on the flip side, if you think about what's happening during active tuberculosis, one of the ways we've gone around asking that instead of using inbred mouse strain is to use an outbred mouse strain. So for folks who don't use outbred mice all the time, it really is that they've taken the 8 parental strains of mice, crossed them in such a way. Many of you may have heard of the collaborative cross strain, but those collaborative cross strains are now crossed randomly so that now you have a broad group of mice that are genetically heterogeneous, and they are called the diversity outbred mice. So they're genetically diverse in such a sense that the heterogeneity across the population is maintained. So if you infect such a diverse genetic group of mice, the expectation that we had going into the study, and this work was done by Radha Gopal, who was a fantastic postdoc in the lab at that time, is now an assistant professor in University of Pittsburgh. She showed that, in fact, when you infect mice, you do get a spectrum of tuberculosis, as you see in the context of human TB. You can see that mice that form those tight structures have less TB. If you go across the end of the spectrum, mice that have a lot of lung disease certainly have severe TB. And she could go on quantitate the B cell follicles and validate these studies at that time. But at the same time, another student in the lab, Letitia Monin, who also, by the way, just started her lab in England, she was able to show that, in fact, when you go across and look at the mice that had the most severe disease and see what are the top 10 genes that come up, you can see you have a classic signature of neutrophil and type 1 interferon. And this is very similar to what you would see in the context of human tuberculosis. I'm going to draw your attention to 2 of these genes, S100A8 and A9. This is otherwise known as calprotectin. It's 40% of the weight of a neutrophil is this heterodimer. So really asking the question, can we go from our results in the mouse model that showed high levels of expression for S100A8 and A9, can we take that data and look in the serum of human tuberculosis patients and see if we're seeing increased expression of these 2 markers in their serum? And this work was done again by Radha, and this data is the data that's shown here is done from a cohort in Mexico, but we've done the exact same studies in South Africa and India with exactly the same results. And what you can see here is if you look at healthy controls of patients with active tuberculosis and compare them with latently infected individuals, you can see high levels of this heterodimer S100A8, A9 in the serum of patients with active tuberculosis. And what was really exciting for us is you can actually measure the level of this protein in the periphery and be able to assess that there is a good correlation with the amount of lung damage that we measure by a chest radiography in these patients with tuberculosis. So here's another example where we're able to take a discovery in the lab, move it along to translational space by confirming that humans are expressing this protein. And in fact, Nilisha Scott, who was a student in the lab, went on to show in a recent paper in JCI that this gene expression comes out 200 days before the patient's able to have sputum positivity for TB. And Nilisha, who's shown here in this picture, was able to then go back and look at the S100A8, A9 knockout mice and show that, in fact, these mice better control infection during chronic infection of TB. Shown here, you can see day 100, 150, or 300 days post-infection. These mice have lower lung burdens of TB in their lungs when compared to the control mice. And then coming back to what we did in the context of single cell sequencing in the monkeys, we were again able to see that this S100A8 and A9 that's shown here was one of the highly expressed genes in this macrophage population that had interferon signature in the context of macaques. But also when we looked for these interferon responsive macrophages and measured them in the periphery, we could see that there was a perfect correlation with lung pathology in patients with pulmonary TB in a cohort in Mexico. Again, suggesting that when we see inflammation, TB disease, we're seeing markers of type 1 interferon. We're certainly seeing S100A8 and A9 as key features of those markers, and neutrophils as well as macrophages seem to be expressing it in the context of infection. We then did a study again spearheaded by 3 different people in the lab, my lab as well as Macadonka Mitriva's lab, where we compared cross species to see if there was any value to what we were doing in the mouse. And we found that, in fact, the mouse model very well correlates with the expression of TB disease in macaques. or the expression or across species to human tuberculosis, allowing us to say that the mouse is a good tractable model to study chronic TB inflammation. So just to put a lot of data that I've shown you in the past few slides so I can move on to the vaccine studies, really what I hope you leave today with is that all TB granulomas are not alike. Depending on whether you have TB disease or whether you're actually protected from TB, the lungs, the immune responses in the lungs are driving very different flavors of granulomas. In one case, you're seeing these follicles that are forming that are very tight, that are mostly lymphocytic in their nature. They could be innate lymphoid cells or adaptive lymphoid cells, and they all have a very unique but interdependent role in controlling MTB infection. On the other end, work that we've done, but several others in the field have gone on to now give us a better understanding that we're really seeing a myeloid cell infiltration in the context of active tuberculosis. It's a very classic type 1 interferon signature that we see that's associated with TB disease progression in the context of active TB. And of course, as I've shown you, we've used data across different species to really come to this conclusive slide. So the next maybe 5 minutes or so, what I want to leave you with is some ways that we can take the biology and the pathogenesis that we've understood from our bench research and how do we translate this now to make new vaccines for TB. One way you could think about this is people who form these structures seem to be protected against TB. Can we use vaccines or can we use vaccine strategies to induce these lymphoid structures in an attempt to really make new and novel vaccines for TB? And that's really, again, this has been published. I've shown this data before, but just to remind you that what we see during TB latency is this high level of formation of these lymphoid structures. So it appears that when TB is controlled, you get a very good immune response to form these structures. So one way to think about a new vaccine strategy for TB is maybe we can make a mutant of TB that is not, that is attenuated or not infective. And will that induce these immune responses the same way it would in the lungs of latently infected individuals, or in this case, macaques? And that's exactly the experiment we did in collaboration with Deepak Kaushal's lab. CGH is a gene in TB that's a master regulator of in vivo stress in the response of MTB infection. When mycobacteria do not have the CGH genes, when you infect macaque lungs with CH, the MTB is attenuated in the context of infection. And so here's the data that we publish, showing that if you take a wild type TB and infect macaques, as you can see here from the symbol, with MTB infection, you form these small structures, and it's very similar to the data I've shown you before. But when you take MTB-CGH, which is a mutant that lacks the CGH master regulator, you now form large structures of B cell follicles, and that's measured and shown here. Of course, if our hypothesis is correct, and these B cell follicles are providing protection, we should be able to now go back and challenge these monkeys with a lethal dose of TB and ask the question, are these macaques better protected against MTB infection? That's exactly the experiment that's described here. So we use BCG aerosol vaccination as a comparative to MTB-CGH, and we have our controls, which are unvaccinated monkeys. We are vaccinating them 8 weeks later. We are challenging them at MTB, euthanizing them, and looking at CFU levels. This again is published data, so you can look over the paper. But really, you can see that while mucosal BCG is doing a small effect of protection at week 13, and we are showing here CFU measurements for the per ml of the lung homogenate, you can see that monkeys that had induced these fantastic eyeball structures the MTB was controlled when we measured it compared to BCG or the unvaccinated, suggesting that this is one strategy for us to use to go on to forming these structures. And if you look histologically at the lung, here's unvaccinated lungs. Again, you see those large necrotic cores because there's a high dose infection. You can see that in the context of BCG vaccination, perhaps the lung is a little bit better protected. But if you gave an MTB-CGH vaccination, not only are you seeing complete control of MTB, but you're seeing the classic features of what we call as eyeball structures in B cells that you can actually stain here. And you can see that the red staining is CD20 staining, and that's well reflected here in the context of macaques that have received MTB infection, gone on for a challenge with MTB. And then, of course, we come back and say, are the structures really the ones that are providing protection in this model? So most recently in the paper we published last year, we went in and gave anti-CD20 antibody to deplete those B cells within those MTB CGH vaccinated macaques to ask, do they have a role for protection against TB? And the data we published last year show very clearly that when you now deplete B cells in the context of those follicles, the monkeys now have higher CFU of MTB. So you're not seeing the full control that you see in the isotype control on the previous slide, but you're seeing absolute control with loss of the granuloma-associated structures. So this is one way that we're thinking that we can induce and use as a vaccine platform, is to make mutants of TB that can induce eyeball structures that can be used as an attenuated vaccine for TB. The second example I'm going to give is delivery of vaccines mucosally, where there's been quite a bit of work showing that delivery of mucosal vaccines primes dendritic cells and allows dendritic cells to prime T cells and B cells that are then recruited back to the mucosal surface. And here we are working with Blue Willow Biologics, where we have developed a vaccine, which is a nano-emulsion vaccine, which encapsulates MTB antigens. And Dr. Mushtaq Ahmed in my lab is organizing and leading these studies. And he showed that, in fact, this nano-emulsion vaccine, when compared to BCG, does induce Th17 response in the lab, shown here by ELISpot measurement of Th17 cells, either in the lung or the spleen, whereas BCG drives predominantly an interferon gamma response. What Mushtaq was also able to show was that when you challenge these mice with MTB, you can see that this challenge really is similar to what you would get with a BCG vaccination, where the unvaccinated mice have about 5 logs of bacteria. BCG, either alone or in combination with nano-emulsion vaccine, TB vaccine, now protects upon MTB challenge. And I think the real curious data here is if you give BCG along with these nano-emulsion vaccines, shown here in the below graph, you can see that in green is the B220 staining for B cell follicles. You really amplify these follicles in the lung as measured and also shown here in the average size of the B cell follicle. So this is another way that we really are thinking about moving this vaccine to the next step, where we can now use this in combination with BCG to really drive these immune responses in a way that we can give it mucosally, and certainly is one of the ones we are moving forward with next steps. I'm going to skip this slide and move to my conclusion slide, really to leave you with the message, hopefully, that I've shown you is convincing enough to really understand that not all TB granulomas are equal. We have to think about the clinical stage that the individual is. If the individual is really controlling MTB infection, then predominantly they have mounted a very efficient lymphoid immune response. This could constitute both an innate as well as an adaptive immune response that is efficiently mounted. And that's what's really controlling MTB infection. Obviously, at the other end of the spectrum, you can imagine when MTB has really now established infection and the host has lost some control of infection, the flavor of the granuloma is predominantly a necrotic granuloma that is well represented by myeloid cells. And this could be both neutrophilic as well as macrophages that are expressing type 1 interferon that really form, and certainly PDCs too, as new the data from Russell Vance's group and others, that are really driving active tuberculosis granulomas. And obviously, I'm sure many of you will recognize that what I've simplified is a very simple reflection of the large spectrum of TB, and you can imagine a variety of different circumstances in which these granulomas could be very, very fluid across these 2 different types of infection. And certainly, as I've just shown you 2 examples, we are very much interested in trying to modulate these immune responses to benefit vaccine design, limit TB pathology, and certainly an important part of what we're doing is how is TB driving the shift between these 2 different ends of the spectrum? What is their role in making this decision, whether it's going to be a lymphoid granuloma or a myeloid granuloma? I think with that, I'm going to leave this last slide up. This is a picture of our previous group that came to University of Chicago to visit in the summer of 2022. So with that, maybe I will just end by saying, as we reestablish the new group, we are always looking for postdocs, technical staff, and staff scientists. My email is up there. Feel free to email me about any inquiries. I think I did okay on time.
Perfect.
Okay.
Well, Shabaana, thank you so much for this really elegant and very inspiring lecture. I think you made a very good promotion for joining your lab. And again, very happy that we had you here. I feel, of course, now everybody feels probably tempted to ask questions. I'm sure there's many questions, and I could do so as a speaker, but I think we should stick to our rules, and I would refer everybody to the former Twitter, now X page for asking questions. I'm sure there will be many questions. And with that, I'd like to finish our session today. Thank you all for joining. I hope you will join again next week when our speaker will be Hiroshi Ono. And with that, I wish everybody a nice evening or a beautiful remaining day. Thanks again and see you soon. Bye-bye.
Thank you all.